All right, so chapter 14 is going to be about Mendelian genetics, and what we're going to talk about are the Punnett squares and just how things are inherited, and we're going to tie it in a little bit with the meiosis and things like that we talked about. So the main idea is here that you, as a living organism, you're going to inherit what you need from your parents, and what you get is kind of half your mom's, half your dad's, and the inheritance of these genes follow certain patterns, okay? So one of the TOK things that I want you guys to kind of take a second and write something about is there's a link between sickle cell anemia and the prevalence of malaria. How can we know whether there is a causal link, a causal link in such cases, or simply a correlation? Okay. And then the other TOK thing that I want you to write about says Mendel's theories were not accepted by the scientific community for a long time. What factors would encourage the acceptance of these new ideas by the scientific community? So take a second and write something about those two TOK things, and I might collect those as an assignment, or we might just talk about them. So just make sure that you get those done for me, okay? And have them maybe done on a sheet of paper where if I need you to tear it out, you can, or print it out, you can, something like that. So just make sure you get those things done, all right? So now there's a couple of important terms that I want to discuss with you. One is about the genome. The genome is all of the genetic information in your body, and a gene is the kind of heritable factor that you're going to inherit from your mom and dad. It's a piece of DNA that really is going to influence character in your kind of phenotype, and it's just a specific kind of string or sequence of A's, T's, C's, and G's. And the way we know a lot about the genes that we have is because of the Human Genome Project, and that was something that was kind of conceived in the late 80s, and then it was begun in 1990, and then took about 13 years to get nearly fully complete. So that's the Human Genome Project. So we said a gene, this is going to occupy a certain spot on the chromosome. And of course, if you look here, you can see that the allele for purple flowers is right. Whoops, sorry, got the wrong thing here. The allele for the purple flowers is going to be right here. Okay, and then you can see that the allele for white flowers is right there. So they're both in the exact same spot on the same exact chromosome, and they're both, in this case, influencing flower color. Okay, so a character is something that you inherit, like flower color, for instance, and a trait is a variation of that character. In other words, purple flowers versus white flowers or something. True breeding, this is something where you would have maybe, say, big B, or you could say little b, and those would be, homozygous, of course, either dominant or recessive, and there's no variation in the trait there, okay? So hybridization, this is where you cross two variants of two true breeding. So if we're going to do a Punnett square of this, if you had two true breeding plants and you had, say, big B, and little b, of course, if we did these, you'd have big B, little b, big B, little b, big B, little b, and of course big B, little b. And these are all considered to be hybrids. And by the idea of the genes from both parents kind of coming out maybe in the next generation, you're seeing them there. And just depending on the degree of dominance, which we'll talk about, they're probably all going to have the big B phenotype. But when these hybrids cross, you're going to have, say, big B, little b, and big B, little b. This is where you can get some of the parental varieties back. Big B, big B, big B, little b, big B, little b, and then of course little b. So this would come out in the second generation there. Okay, so that's just some Punnett square stuff, which I'm sure you're familiar with. Now, some of the important terms you want to focus on: the P generation, this is the parental, this is usually what starts the whole thing. So if we go back here, if we consider this the parental generation, we could maybe put like a P around that would be the parental. The F1, this is the first filial generation. In other words, this is the first set of offspring that came from the parents. So if we look here, these, for instance, would be the F1, and then this would be the F1 cross up here, and then these right here would be, of course, the F2. And the F2 is just the second filial generation. This is the offspring where you cross the hybrids or something like that. So this is where you're starting to see these traits that we were talking about here. The traits are reappearing from this P generation. And again, if we go back, here's our P generation right here. These traits, when we cross the hybrids, start to show up in this F2, again there, and then here. But you would see this as these three right there. Okay, but especially this would be one of the traits that we would see that looked like that parent. Okay. So now we have homozygous. These are traits where everything is identical. We've already went over this, big B, say, or little b. A heterozygote would be big B, little b, for instance. And then the phenotype, this is the outward expression, what you look like, okay, the outward appearance of the organism. And the genotype, of course, is the genetic makeup of the organism, what the actual genes say. And those aren't as easy to distinguish necessarily as the phenotype may be. Okay, so Gregor Mendel, this is a person you've probably heard of, and he was an Austrian monk, and what he did was he did lots of experiments with peas, and he carried them out from the pea generation F1, F2, and was able to kind of figure out a lot of the patterns of inheritance. Okay, so these are some of the traits that he kind of looked at: flower color, flower position, seed color, seed shape, pod color, stem length, these types of things. But you can see that there's a lot of data there that he kept. And what made his work so good was that he kept excellent records of the things that he did, results of his experiments, those types of things. And at the time, there was a lot of people that believed in this idea of blending, and it makes perfect sense. But when you start really thinking about it and looking at it, like we said, a lot of people were uneducated at the time and didn't really look much farther than the things that were in front of them. And then some scientists, of course, did, and they figured out a lot of things that you kind of casually might have thought made sense actually didn't. For instance, with blending, if you cross, say a purple and a white flower, you might get a lavender flower, or a red and a white flower, you might get a pink flower, or something like that. And people thought the traits kind of blended together, and in some cases, it really appears that they do. But a lot of Mendel's experiments kind of did away with this notion because, for instance, with the purple and white flowers, when he crossed two purple flowers together, for instance, he was able to get, in this case, some of the white back. Okay, so if we look here, here's some of the white flowers that he was able to get back. And just as importantly, if he were to cross big B with little b, you'd think that, for instance, if this was a purple flower, okay, and if this was a white flower over here, that these big B, little b, all four of them might be, say, lavender or something, okay? And I don't know if that's spelled right, but they're not. And then when you cross the hybrids here, you can see that this one here and this one there, those are both hybrids. You get some of this parental variety back there. So he was able to determine that purple was dominant to white. And so he kind of wiped out this blending hypothesis because there was no pale purple or lavender types of flowers. And he also gave rise to what was known as a heritable factor. This kind of term is what he used to describe it, and we refer to these as genes, of course, now. And there must be some sort of a heritable factor that's controlling the flower color, according to Mendel. And so he came up with a 4-part model to kind of explain what he what he saw. In one, there's an alternative version of genes, and he called those alleles. And then each organism inherits a copy of an allele, one from each parent. And then some alleles are dominant while others are recessive. And 2 alleles segregate independently from one another during meiosis. Okay, so the alternative versions of genes, for instance, if you look at this, you can see there's a purple colored flower and a white colored flower, and they're each governed by the presence or absence of the alleles here. Okay, and in today's terminology, kind of what we would say is that a gene resides in a specific locus on a chromosome, a specific spot, okay, called a locus here, or sometimes when you're referring to multiple ones, you might see LOCI, the loci. And the DNA at that locus varies slightly, sometimes just by one base pair, and that's what can kind of make the difference between the phenotypes. And so that gives you the different genes. And the different sequence, of course, is what's changing the flower color in this instance, or any of a number of other traits. And these alleles are due, again, just to little variations, mutations give rise to these alleles, etc. The second part, for each character, an organism is going to inherit 2 heritable factors, one from each parent. And Mendel knew nothing about what the cytologists were studying with regard to meiosis at the time. So he didn't know anything about like haploid, diploid, the ploidy, or anything like that, or anything about chromosomes. He just kind of deduced that you must get one thing from one parent and one thing from another based on these experiments that he did. And of course, we now know meiosis produces haploid cells which combine to form diploid organisms, and the alleles are carried in these haploid gametes, okay? So if two alleles are different at a particular locus, then the dominant allele is going to determine the appearance, while the recessive allele is kind of going to get masked and you're not going to be able to see it until maybe the next generation. Okay, and the two alleles for heritable characteristics now segregate independently from one another during meiosis. And we also know now how meiosis works and why, when they line up at the metaphase plate, how they can align one way or they can align another way or they can align, a variety of different ways. And this is what makes up the law of segregation. And what it does is it applies to all the genes on a particular chromosome and says that the alleles are going to segregate independently from one another, regardless of what phenotype they're carrying, just as long as each gamete contains one copy of each trait. And in terms, again, of the chromosomes, just recall that homologous chromosomes are being separated out during the process of meiosis, and those gametes are going to kind of contain the different alleles that are being distributed during this process. And these figures here aren't very big, but they're something that you can look back to from meiosis in the meiosis lectures, because we've already looked at those and they're also in your textbook. Okay? So now when the gametes fuse with one another, what you're going to get, of course, is that diploid zygote back. And the results may be that the two alleles are the same. They might both be different. It doesn't really matter. It's just that when they fuse, you're going to get that diploid organism back. And if the alleles are present, if different alleles are present, I should say, you're going to have a 50-50 chance that the gamete's going to receive one allele or the other. So in other words, when you're looking at this, these are both the same. So obviously, these are only going to produce this type of gamete here. But when this type of an organism gives rise to gametes, one is going to be big P, the other one is going to be little p, and you've got a 50-50 chance of which one you get. And it's just a crapshoot. So if you were to have 100 offspring, there's a good chance that 50 of them would get the big P allele, and then the other 50 would get the little p allele. Okay, and we'll go over some of that with our lab and things like that. So if the alleles are the same, again, we said that each gamete is going to contain the same one. We just went over this, and we're saying that these ones up here, these would be true breeding, okay? And then these ones down here, of course, are going to be the hybrids, okay, or heterozygote. So the law of segregation using a dihybrid cross, Mendel was able to show that the gametes are going to receive, these alleles from their parents kind of independently of the other genes that are there. So what he was looking at was he had yellow round seeds, and he made flowers, made plants, and these were true breeding ones, and he crossed them with the green colored and wrinkled types of seeds, and I'll show you a Punnett square in this in a second. And if they assort independently, you're going to see a 9:3 to 3:1 ratio. All right, and that's kind of characteristic of a dihybrid cross. And if they don't assort independently, then the ratio is going to be something other than a 9:3 to 3:1. So if you look up here, you can see with this, there's only one type of gamete that it can produce, and it's right there. And with this, there's only one type of gamete that it can produce, and it's right there. So these organisms, when they combine, are going to give this. Now, these can form 4 different types of gametes, and see if you can figure that out here, okay? And just look at it and see what types of gametes you can produce. And remember, meiosis produces haploid gametes, so these are diploid, okay? And then these contain one Y and one R, so these are haploid. So see if you can figure out what these would be before I go on. And if you looked at it or if you thought about it, you're going to get one gamete that's going to be big Y, big R, one that's big Y, little r, one that's little y, big R, and one that's little y, little r. And you can see that down here. This one and this one represents these gametes right here. Okay, and then when you do the cross and cross these up, if they're assorting independently, you're going to see this 9:3 to 3:1 phenotypic ratio, and that's exactly what Mendel was able to show. Okay, he concluded no matter how many characteristics there are, they're always going to segregate independently of one another. It's going to be a crapshoot as to how these gametes kind of combine. So if these are the two gametes that are produced, they're going to produce offspring that look like this, okay? And so all of these yellow round seeds are big Y, and it doesn't matter what the second one is, and big R, doesn't matter what the second one is. And we'll go over this more in class, okay? So as far as the 3:1 ratio goes, the law of segregation accounts for this as well. When you're using this Punnett square, it's easy to see, and you can examine the ratios or what are determined here, just like you could with the dihybrid cross. This is the mono. These things are going to align, and this is your 3:1 ratio. 1, 2, 3. to 1. So there's your 3 to 1 phenotypic ratio, 3 to purple and 1 to white. Okay, and we can also show this using a test cross. And what a test cross is a cross to the homozygous recessive. So if we have a purple flower, okay, we don't know if this purple flower is big P, or big P, little p. We don't know which one it is. But if we cross it to the homozygous recessive, in other words, if we cross it to little p, we can figure it out real quick. If all of them are purple, then we know that this had to be what the parent was. And if any of them were white, then we would know that it has to be a heterozygote. Okay, and it would produce about a 1-to-1 ratio. So if we look at the crosses here, we want to know, again, is this big P or big P, little p? If it's big P, here are the two gametes, and when we look at them, all of these are going to be purple. And if we look over here, if it's a heterozygote like this, in other words, big P, little p, then you're going to have a 1-to-1 ratio, or about 50% of the offspring are going to be purple, and then about 50% are going to be white. Okay, so that's how the test cross is used. So if you have any questions, we can definitely go over that in class. All right, so what we'll do now is we'll pick up on the next lecture here with the law of independent assortment. All right, so we'll talk to you soon.